Okay, internal lamprey features. So at this point, you should have your sagittal section complete, and you might have a male and you might have a female. It seems like on average we receive more females than we do males. So you probably have a female, but you might have a male. If you have all these eggs everywhere, clearly have a female. If you don't have these eggs everywhere, it's a high chance you have a male, and you will see the reproductive organs in the same area, specifically the testes in the same area here, and they just look like glandular tissue. So because these eggs are so messy, I often like to try to get them out of the way first so that I can see everything else better. While we do that, we should actually look at them. So I'm going to zoom in so we can see them a little more closely. Okay, I'm going to work with one section at a time. So these are the individual eggs. Obviously, this particular animal would be more of an R strategist, where they produce lots and lots and lots of individual offspring and hope that a few survive. So this one female can produce thousands of offspring, but not many of those offspring will actually survive to reproductive age themselves. Okay, so when you're looking at this, the eggs are easily removable here. You can just brush them right off. The ovaries themselves is this tissue. So you have the ovarian tissue that's producing the actual eggs. And they're little hard things. They're just everywhere, messy all the time. So you can use your finger, now that you know ovary versus egg, and you can carefully start removing them. Pull them up. Don't just start yanking them out, because chances are high you will end up destroying something else. You don't have to remove all of it. You just want to get enough of it out of there that you can see everything else a little more clearly. Okay, so now I've got most of that ovary gone. I want to remove the eggs. Be careful when you see things like this. Don't just say, oh, well, surely that's nothing, and because it turns out that it's actually something. So only remove what you're sure you want to be removing. And if you have questions, ask me. If you're not sure if something is important or not, and I will answer. Okay, so eggs everywhere. So what I'm going to do now is I'm going to actually take this over to the sink, and I'm going to very gently rinse it off to try to get these eggs clear so that I can see things better. Okay, so it doesn't take long, and it's worth it to be able to see better. See how that's much more clear now? All right. Let's take note first of the inside wall here. Now you can see these individual myomeres very well. Individual myomere, each muscle segment. You need to be able to identify those. Okay, start up here. Start looking for features. Even my probe is full of eggs. To be totally honest, I prefer dissecting males. You don't have to deal with all these eggs everywhere. At any rate, this stuff will be the same male versus female, so this will apply to both, whether you have a male or a female. So just start in the head and generally orientate yourself here. You can see again the teeth, the mouth itself. This muscular structure here is the tongue. It ends here with this, the horny teeth there. So tongue. Trace things with your probe, okay? So if you think about what the blood meal is going to do, it's going to go back into here. And see how this is just a cavity there? So mouth, pharynx. The pharynx will lead to the esophagus. If I trace it carefully, I can see here. Esophagus. Okay, the esophagus is an actual tube. See how my probe starts to disappear there? If I very carefully insert my probe along there, it would be gentle because you don't want to tear too much stuff, but you want to get an idea where it's going, right? You see it there. So it's through the esophagus here, and then the intestine. So that's what this is the intestine. Intestine running all the way along there. And if you trace it all the way down, it's going to drain right out the cloaca, because that's where the animal will release waste. Oh, I just got her back in her own eggs again, so I got eggs everywhere again. So if you go down here to this end of the digestive tract, and you very carefully insert your probe. Now that you want to get the papillae out of the way, you don't want to insert it in the papillae, not at this point anyway. But if you're lucky and you do it carefully, there, you can see the probe through there. You can actually see that it comes into the intestine right there. Okay, so this is how it's going to work. Blood meal, mouth, esophagus, or pharynx. Oops, gotta get where you can actually see this in the frame here. So, mouth, pharynx, esophagus, runs dorsal to the gills here. And then right about here, this is the heart, and right about here becomes intestine, and where the digestion is going to occur. Intestine runs all the way down and out the cloaca. So once the blood meal has been digested and the nutrients absorbed from it, then the waste will be eliminated through the cloaca. Now if you're lucky, when you have the intestine, you can keep it relatively intact. You can very carefully create a little slit. It doesn't have to be a big one, just a little one, so that you can get your, I think I just lost track of my little slit there. Dissecting scissors in it. And then you can cut along the intestine, because it's a tube, right? I mean, that's what it is. So like you're cutting open the tube, and then actually examine the interior of it. And you'll notice that it has very small, very, very small ridges. See, I don't know if I can get this to focus on it well or not, but let's see if I can. Focus. Nope. That's what I get for using an old camera, right? Okay. Zoom back out, let it focus again. Okay, well, you look at it when you actually open it. Notice the very small ridges there. Okay, that's going to increase surface area. So what does the intestine do? Chemical digestion. Surface area is key. The more surface area you have, the more efficiently you can actually do the digesting. And you will see as we go along in the complexity of organisms, the complexity of those ridges increasing too. Okay, now what didn't you see? Should have noted the lack of stomach. Why doesn't this have a stomach? Well, what does the stomach do? Mechanical digestion. which means it churns up solid food and makes it mushy so that you can actually get it chemically digested in the small intestine or the intestine. In this case, they don't eat solid food, so there really is no need for a mechanical digesting stomach. So they don't have a stomach. Okay, so there's the digestive system. Let's look at the notochord next, because the notochord is actually going to be a great reference point for everything else. So the notochord is this structure that's kind of soft, it's flexible, thick, uniform in color and appearance. It's the most obvious structure running down the midline of your animal. That notochord is essentially what its endoskeleton is. It's not bony, right? This is not an osteichthyes, it's a cephalosporomorphy, right? So it's not bony, but it does provide structure. Why? Well, when muscles contract, and you can look at this on the PowerPoint, it'll give an explanation with some diagrams and drawings, but when muscles contract, they squeeze, right? And if you don't have a structure for it to squeeze against, the animal would just collapse. So what this does is provide enough support that it has something for the muscles to actually work against without collapsing the animal altogether. Okay, so use the notochord to orient yourself for everything else. So you should find the major blood vessel and the intestine should be ventral to the notochord, whereas the spinal cord and the hemopoietic tissue is going to be dorsal to the notochord. Do not, on a test, this is a very common thing for students to mix up, do not call that the spinal cord. It is not. This is the notochord. Okay, a good way to tell that is it doesn't attach to the brain. So if you go back up to the head end, brain leads to spinal cord, right? Now, these aren't the most intelligent animals in the world. They don't need to be in order to survive. So their brain isn't very large, but it is there. You can find it just posterior to this black structure, which we'll talk about in a minute. This is the brain. Okay, the brain is in the chondrocranium, this little cavity here. It's got a little bit of cartilage protecting it. And then it goes from the brain in this canal. This little canal-like structure runs along that. It's just dorsal to the notochord. This is where you're going to find the spinal cord. Now, the spinal cord is very small and thin, so it's easy to destroy. Sometimes you'll get lucky and it will come out perfectly in your dissection. Sometimes it won't. That's a little piece of spinal cord there. As I travel down, I'm seeing mostly, well, the eggs that just went everywhere. And then down here a little more, I start to see where, just luckily, maybe the spinal cord is a little more intact. There we go. Here we go. Spinal cord more intact down here. So I can get my teasing needle under it, and then I can very gently just try to pull it up a bit along the line there. You don't have to do it the whole way. You can. I mean, why not? As I see, you've always got time. We've got lots of time to do these dissections. You don't have to be done in even a couple of days. You've got plenty of time. So take your time and see how well you can get some of these things dissected out. At any rate, that's the spinal cord, and it's going to run all the way down the animal to the tail end here. And it will run all the way up and connect to the brain, which is here. And you can remove the brain if you'd like. On sketch, you're going to need to know some basic parts of the brain. So make sure as you're going through your notes and your work, you've identified that. And you can look at it and try to see if you can identify any of them. They don't distinguish super well here, but if you want to give it a shot, which I would recommend doing, put this under a dissecting microscope, and then you can see the individual structures a little bit better, and you might be able to identify some of the structures on more than just sketch. Okay. Eggs everywhere. All right, so let's go back to this olfactory sac here. Olfactory sac. I always said that wrong. I don't know why. It's in my head to say it wrong, but it is. Anyway, so if you look externally here, you've got the median nares. Like I said, the median nares will lead to the olfactory sac. Okay, so there. See, if I go with my probe now, so I said not to put your probe too far into that median nares when you were looking at it externally because you didn't want to trash the delicate structures on the inside. Now, and teasing needle sometimes works better than a probe for this because it's smaller. It has a smaller diameter. But now you can actually see that that will lead down to the olfactory sac here. And you can go both directions. And I didn't make that hole, that was there. So the water will enter and come into this olfactory sac. And look at it closely, and you'll see it's got lots of infolding. Again, it's a surface area thing. Surface area becomes very important in a lot of things with anatomy. If you're absorbing something, or you're exchanging something, or you're sensing something, surface area, increased surface area, will make that more efficient and more acute, and will provide a selection advantage, a reproductive advantage in the case that you're able to sense your environment better or digest your food more efficiently or whatever that is. So anytime you see those kinds of infoldings and extra surface area, think about that in terms of what advantage that selective advantage that surface area would provide. Okay, so sense of smell. Throughout the head region, you'll notice there's these occasional structures that are harder. These are the cartilage structures, and it's all in this big basket, all interwoven throughout the soft tissue there. That is a support system of cartilage. I'm not going to make you know all the individual parts of that, but if you do see a harder, shinier cartilage area here, make sure you can identify that as cartilage and that it has support structure behind it. Well, let's go back to the olfactory sac for a minute because I forgot to tell you about the hypophyseal pouch. Okay, so right underneath, so just underneath, of course in this case means ventral too, the olfactory sac, you've got this cavity. Now you might think that means nothing. You should be wrong. So this cavity here is actually serving a really important purpose. This is the hypophyseal pouch. So it's got muscles around it, and what will happen is it will inflate and deflate here, which creates some negative and positive pressure in the olfactory sac that makes this olfactory sac work like a bulb to get water in and out in order to sense the environment as it goes. So as this bulb is squeezed, okay, it'll shoot the water out, and then when this inflates, this pouch here expands again, it will pull this with it, which will widen this, which will inflate this area a bit, this olfactory sac, and that will create an area of low water pressure. Now, low water pressure relative to what is just on the external side of this lamprey here, this will be an area of high water pressure. This right in here will be an area of low water pressure. And what happens? Diffusion. So that pressure, the high pressure, will move toward that low pressure area. The water itself will move from that high water pressure area to the lower water pressure area, and it will bring water inside that. And then again, this will contract. When you contract that, if you remember from basic physics, when you contract an area and you make smaller that volume of liquid, you increase its pressure, right? So now I'm going to increase the pressure inside this olfactory sac. It's going to create high pressure in here relative to what's just outside of the median nares there, and it will actually then force the water out to where now is relatively lower pressure. And then it'll expand again and bring in more water and squeeze again and push the water out. remember that system, because when we get to learning more about about land vertebrates and their respiration, that system will come in handy to understand it now, because your breathing, our breathing, actually works in a very similar way. Okay. So then, from that, let's talk about respiration. Okay. Now, they don't use their nose for respiration like you do, correct? Their median nares, their nose, their nares is purely for smelling the water. These are full of chemoreceptors for smelling the water. What are they actually using for respiration? They're using their external gill slits to bring in water into their internal gills. All this feathery stuff, internal gills. Oops. All this feathery stuff, internal gills. Okay, so take your probe, find an external gill slit, and gently push your probe through. Gently. Okay, it's going to come out in here on the inside somewhere and through. Oh, I went out of the frame again. Okay, through. Okay, you can go down further, and sometimes, if you're careful, don't force anything, just work it carefully until you come out somewhere internally there. Okay, you can also do that here. See, now if you open one of these gill pouches here, you can see what appears to be a little donut shape. That is the internal gill slit. It should lead directly to an external gill slit. Okay, internal gill slit, external gill slit. It's like different sides of the same coin. So water will come in. It'll come into this larger pouch area that will hold the water, and that's where that water can come in contact with all these gill filaments. The gills themselves are made up of all these filaments. They look kind of like little bitty fin rays, but they're not nearly as stiff because they're not the same material, right? What are these? These are the gill filaments. Feathery appearance provides lots and lots and lots and lots of surface area. Imagine if this were just a flat surface. There wouldn't be nearly as much as creating extra surface by making it into filaments. And all this filamentous area is for gas exchange. Again, that works through diffusion. So, diffusion, things go from high to low concentration. When it brings, quote unquote, fresh water in, what has happened now is these gill filaments will be full of blood that has come from other parts of the body and is carrying a relatively high concentration of CO2. That CO2 then will be a higher concentration inside these filaments than it will be in the water that has just come in. So what is that CO2 going to do? Remember, the CO2 is small, so it moves readily through the cell membranes because it's small and non-charged. So it goes through the phospholipid bilayer. Remember, back to biology 1 for that. And that CO2 will move readily from the gill filament into that quote-unquote fresh water, making it higher concentration until it reaches pretty close to what would be in equilibrium, and then the CO2 will stop transferring, but there'll be much less CO2 in the blood at that point. The same applies for the O2, the oxygen. So the water coming in will hopefully be well oxygenated water, and there'll be plenty of oxygen in it, and this blood in the gill filaments will have less oxygen in it relative to the water. So if there's more oxygen, higher concentration relative to the gills in the water, what will happen? That oxygen will go from the water across the cell membrane, because it also is small and non-charged. It can get through the phospholipid bilayer, across the cell membranes, into the bloodstream. And then it can be carried throughout the animal to be delivered to individual cells that need it in order to perform cellular respiration. Remember, back to biology 1 again, cellular respiration, oxygen acts as the electron acceptor for the end of the electron transport chain. The electron transport chain is where most of that energy and ATP is made. You need the ATP to do everything else, right? And if you aren't using the electron transport chain, you're not creating very much ATP as you use your sugar through glycolysis. So That's the difference between aerobic oxygen and anaerobic no oxygen respiration. These animals, because they need lots of energy, have to have that O2 as an electron acceptor in the electron transport chain in order to create enough ATP to sustain themselves. And they get that from the water by diffusion through these gills with all this surface area. And what actually is going to pump that water around will be the heart. Now, depending on how you did your sagittal section, you can see here, I went a little wonky right there. Happens, it's all right, no big deal. So I actually ended up with the heart intact. Usually, if you do your sagittal section carefully, you don't. You look at the other side here. That's just part of the pericardium there, or the cardiac chamber there. Okay, or the cavity. Okay, so at any rate, heart. I ended up with it intact. Yours might be intact. It might also be sort of cut in half. It just depends on how your section went. This cavity surrounding it is called the peri-around cardial heart cavity, pericardial cavity. And notice too, it separates the gill area from the rest of the body. That's going to continue to be a theme, and many animals will have versions of that because it existed in our ancestors, right? Okay, so because I have it still intact here, I'm going to go ahead and take it out. The PowerPoint online has the more common section where the heart is in half and you can see the chambers straight off. This is one of those things, again, with dissection, you never know how it's going to go until you get in there and do it. And sometimes you end up with some different things just based on how it goes, so go with it. So I am going to remove the heart in its entirety here, if I can. If you can't do that, that's totally fine because, you know, like I said, yours is likely to be cut in half. Now, of course, the heart is attached with vessels to the rest of the body because it's got to be able to flow through their bloodstream. There's actually a major blood vessel that runs just ventral to the notochord. That's part of what this heart is attached to here. You see right there, this part of this attachment area, that's some of that blood vessel being attached there. Okay. Now, I know as students of the information age, you guys use Google all the time. Be careful about relying on Google for identifying certain things. There's the heart. Because different levels of anatomy will identify these things differently. You might see some places consider this heart to be 3-chambered, other places 2. I'm calling it 2 chambers. Basically, you're going to have an atria and a ventricle. The ventricle is going to be very muscular, and that's true of all animals because that's what's doing the major pumping. That's got to be strong to really create the blood pressure. The atria is usually what collects the blood and then sends it into the ventricle for pumping. So fish, we're saying 2-chambered heart for fish, 3-chambered heart for amphibians, and in the reptiles you start to see the 4-chambered heart. We talked about that earlier when we talked more about chordates. 2-chambered heart, 1, 2. Okay, that's what we're calling this. Now when you get into more advanced kinds of anatomy, they'll break down these individual structures of this one chamber into multiple things and they'll start calling them different things. Don't worry about that. I want you to know at this point in intro zoology that this is one chamber and this is another chamber. Okay, so there's the heart. Most likely, you already have that in sagittal section and it'll look something like that. And you can see the less muscular, sort of like sac-like atria area, and the very muscular pumping section, which is the ventricle. Okay. Now, as far as the circulation, it's going to go gills, delivers oxygen to the body, picks up CO2, travels back to the heart, collects in the heart, and the atrium goes to the ventricle, which is the stronger of the two. So it collects in the atria, it's pushed into the ventricle, and the ventricle squeezes and pushes hard and pushes that blood to the gills, where it can drop off CO2 and pick up oxygen, and then just repeat itself. So it's gills, body, heart, gills, body, heart. Okay? All right. The next prominent thing you'll notice here is the liver. It's green. It's probably larger in males than it is in females. The reason for that being it's used to produce some pheromones, they think, to attract females. So the males have a larger liver, females have a smaller one. It's green because of the chemicals that it produces. But it's this triangular organ right there. That's the liver. All right, moving from that, here, just ventral to the notochord, you can see the major blood vessel there. Okay, so there's a major blood vessel right along there. Probably still has some remnants of blood in it. There's ovarian tissue here too, so I'm trying to make sure I can sort out which is blood vessel and which is ovarian tissue. And sometimes it's hard because I don't always, or I usually don't, order injected lampreys. But if you happen to get lucky enough to get an injected lamprey, the blood vessel itself will be red, and the ovarian tissue won't, or the testicular tissue, in that case, if it's a male, won't be red. So you'll be able to see that pretty easily. And I think I do have an injected one. I thought I saw an injected one. Another lamprey. You'll know because the tail will appear to be broken and have some red area on it. There we go. The places that prepare these will do that and inject it with latex to show you the arteries will be red in that case, and then this might have a slit up top too. I don't have very many of these, so chances are you probably don't have an injected one, and that's fine because they're cheaper. Okay, so there's that major blood vessel. That's going to take the blood with plenty of oxygen from the gills and start delivering it to the body. Next thing you need to know, let's stick with the circulatory system for a minute and look at the hemopoietic tissue. So heme, blood. Orient yourself by finding the notochord. Notochord, dorsal to that, spinal cord, dorsal to that. There's this tissue area here that looks different. It's kind of spotty, it's darker, runs the length of here. That's the hemopoietic tissue, and it produces blood cells. Okay. It helps too if you remember the order these things go in. Okay, blood vessel, dorsal to that is notochord, dorsal to that is spinal cord, dorsal to that is the hemopoietic tissue, dorsal to that is muscle. That will help you on the sagittal, or excuse me, the cross sections, the transverse sections that we will also do later. All right, from that brings us to the kidneys. So, kidney beans are called kidney beans because they're shaped like kidneys, right? Well, they're shaped like mammalian kidneys, not like anything else. So, this is usually something students have a hard time realizing at first. What are the kidneys? I can't find them because they're looking for the little bean-shaped things. They're not bean-shaped. Okay. Kidneys in fish, and lampreys in particular, are flaps, and they're long. So you go mid-ish on the animal, and you'll see these flaps. That's kidney. And it runs down the length just tore some of it. That's okay. Runs down the length of the animal. Here you can see, because I didn't get it exactly midline, you can actually see both kidneys. Often you'll end up with one kidney on one side of one section, and then the other kidney on the other section. In this case, at this particular part, I've got kidneys, both kidneys on this section here, because this is one kidney, and then here's ovarian tissue, and then here's the other kidney. Okay, this is again his intestine. I'm going to move that out of the way. Kidney. It's this dark, almost black color on top. And the bottom of it, the ventral aspect of it here, is sort of like flimsy. The reason for that is this is the archonephric duct. Okay, it runs the length of the entire kidney along the ventral aspect of it, and it's basically a tube. So the kidney will filter the blood and remove wastes, cellular wastes. As it removes those wastes, they'll drain down the kidney and into this archonephric duct. If you're careful, and in these ducts you can see better on the transverse sections, but if you can do this, give this a try. On that flappy part, just take your teasing needle, okay, and very gently just sort of poke it, like you're poking just the top of a tube. And you'll notice that it travels very easily. That's not solid tissue, that is a tube. That's the archonephric duct. And it goes all the way down, and it's down here all the way towards the cloaca. And what will happen down here, okay, so you get intestine, drains out the major part of the cloaca. The pleural peritoneal, we hadn't mentioned that yet. That's this big cavity here. This is called the pleural peritoneal cavity, and you'll hear that repeated again as we go through more chordates. The pleural peritoneal cavity itself, where the ovaries are at and all the eggs, also will drain out the cloaca, because that's how the eggs get out, okay? The archonephric ducts will also do that. So what will happen is both of them will come down until they actually join together, but they, oh, all these eggs everywhere, they drain out this urinary papillae. And if you're really careful, sometimes this is easier than others, you can use a teasing needle and you can find the actual opening. Oh, no, I must have poked right through. And the tissue is very, very, very fragile, so it's easy to not get that quite right. It's easier if you happen to do the cross sections well, then you can trace the archonephric duct right out the urinary papillae. But anyway, so cloaca, the intestine will empty into it, and that empties out of the body. The pleuroperitoneal cavity, where all the eggs are, will also empty out of the cloaca. And then this urinary papillae will project out of the cloaca, and this is where the archonephric ducts will drain into and eliminate the waste that the kidneys have filtered from the body. Okay, and that should be what you have to know for the sagittal section of the internal lamprey. Don't forget to also watch the video for the transverse sections, and I will make some transverse sections that groups can look at individually. If we have enough lampreys, we can make a couple of them. We just don't order enough for every group to have 2, so do not neglect watching the video for the transverse sections, and also making sure that you get the transverse sections at some point during your lamprey study, and make sure you can identify all those too.